You know, what if I told you that making some of the most powerful molecules we need for medicine could be as easy as a one-pot recipe? It almost sounds like science fiction, right? But it's actually happening right now in chemistry labs. So let's get into this absolutely incredible breakthrough. Okay, so the stars of our show today are these things called fused quinolines. And they are molecular Swiss Army knives. They can fight bacteria, they can shut down the hepatitis C virus, they're even used in fluorescent materials. And, most important for our story, they can stop an enzyme called caspase 3 right in its tracks. So yeah, they're kind of a big deal. Because of all those amazing abilities, being able to make these molecules easily and efficiently, well, that's a huge prize for chemists. It's like finding a master key that can unlock all sorts of solutions in medicine. And here you can really see just how valuable these are. In drug discovery, there's this term privileged structural motif. It's just a fancy way of saying that this particular molecular shape is an absolutely fantastic blueprint for building new drugs. Their potential to protect our neurons and especially to inhibit caspase 3, well, that puts them at the very top of the research list. But here's the catch. For the longest time, making these incredibly valuable molecules has been anything but easy. The old recipe was, frankly, a mess. It was inefficient, difficult, and just plain complicated. And wow, this picture just says it all, doesn't it? Up top, that's the old way. Multiple steps, complicated reactions, you often needed special conditions. Then look at the bottom. That's the new way. Simple ingredients, one pot, and you get your final product. It's really the difference between a 3-day cooking marathon and a perfect 30-minute meal. So the bottom line is this. The old ways were just tough. They demanded harsh conditions, lots of steps, and ingredients that were hard to come by. So the challenge, the big question for the scientists here, was could they invent a whole new process that was simple, efficient, and could be done at room temperature right out in the open air with common ingredients you can just buy off the shelf? And the answer, you guessed it, was a huge, resounding yes. They developed a totally revolutionary one-pot method that just changes the entire game. And here it is, the new recipe in all its beautiful simplicity. You take one flask, you mix your starting ingredients, an N-dimethylaniline and N-phenylmelamide, with a little copper catalyst. That gets the first part of the reaction going. Then, and this is the best part, without doing any complicated cleanup, you just add the secret sauce, a mix of silver nitrate and potassium persulfate. And bam, the final molecule just forms. It's that simple. Let's just take a second to appreciate what a massive leap forward this is. One pot means you save tons of time and waste. Room temperature means you're not burning a bunch of energy. Air atmosphere means you don't need any expensive, specialized equipment. And it all works with common chemicals. I mean, this is the definition of elegant, green chemistry. And the results? Get this. The very first run of this new recipe, just done out in the open air, gave them an 88% yield of the molecule they wanted. For this kind of complex chemical transformation, that is an absolutely fantastic result. Of course, you know how scientists are, right? 88% is amazing, but they always want to push the limits. The question became, what is the absolute best case scenario? Could we squeeze out just a little bit more? So they tried it. They removed all the air and ran the reaction under pure nitrogen. And yep, they bumped the yield up to 91%. Now, while that is technically better, the fact that you can get a nearly identical 88% yield without all that extra hassle, that's what makes the open-air method so incredibly powerful and practical. For the real world, it's the clear winner. Okay, so how on earth does this super simple recipe work so darn well? Well, to figure that out, the chemists had to put on their detective hats and uncover the reaction's secret mechanism. They had a hunch, a theory, that the reaction was happening in a very specific way. But a theory is just a theory until you prove it. So, to get that proof, they decided to set a really clever chemical trap to see if they could catch the reaction in the act. And here's the experiment. They added a special molecule called tempo into the mix. Now, tempo is what we call a radical trap. Its whole job is to find and grab onto these super reactive things called radicals. And guess what happened? The entire reaction just stopped. It ground to a halt, and they got almost none of their final product. The trap worked perfectly. It proved that these radical intermediates are absolutely essential for the reaction to happen. So, what even is a radical pathway? Well, you can think of it like a chemical chain reaction, kind of like a series of dominoes falling one after the other. It's all driven by these hyperreactive molecules that have an unpaired electron, which makes them really want to react with something. This new recipe just brilliantly harnesses that intense reactivity to build the exact structure they need. Which brings us right back to where we started. All of this brilliant chemistry isn't just for a textbook. It has a very real, very important payoff, creating molecules that could one day become the medicines that we all rely on. Remember caspase-3? It's this super important enzyme that controls a process called apoptosis, or programmed cell death. Now, that's a normal, healthy process, but sometimes it goes haywire in diseases like Alzheimer's or after a heart attack. So finding a molecule that can block caspase-3, well, that's a massive goal for treating these kinds of conditions. And this is the money shot. What you're looking at is a computer model of one of their brand new molecules, that orange bit, nestled perfectly inside the active site of the caspase-3 protein. It fits like a key sliding into a lock, physically blocking the enzyme from doing its job. And this table right here, this gives us the data to prove it. That glide score is basically a measure of how well the key fits in the lock. The more negative the number, the better the fit. So a known effective inhibitor has a score of about -6.2. Now look at their new molecule, 4C. It's at -5.3. That is incredibly close, and a very, very promising sign that it binds almost as strongly. So what makes it fit so well? Well, the researchers figured out that one specific part of the molecule, this thing called the pyrrolidine-dione nucleus, is doing all the heavy lifting. It's forming these critical bonds inside the enzyme that essentially gum up the works. It's like the teeth on the key that fit perfectly into the tumblers of the lock. This work is just such a beautiful example of elegant chemistry leading to a powerful, practical application. And it really leaves you with a fascinating thought. With simple, brilliant new recipes like this one, what other incredible medical breakthroughs are just sitting there, waiting to be cooked up in a single pot?